Welcome to this video for A-level biology, and in this video I'm looking at testing for starch, proteins, reducing sugars and triglycerides. Before you test for these substances, you need to prepare the sample. So testing for starch, you first need to grind the food in order to increase the surface area so iodine can easily react with the starch. For soluble substances such as reducing sugars and proteins, you will need to first grind the substance, add water or buffer to dissolve the protein or the reducing sugar, and then filter to remove any insoluble substance. It's the filtrate that you will use to do the test. In the first test, which is for reducing sugars, you would use Benedicts reagent, and you can use this to test for monosaccharides such as glucose, fructose and galactose, and you can also use it to test for disaccharides. However, only some disaccharides are reducing, and examples include maltose and lactose. So what are reducing sugars? Reducing sugars will reduce the copper 2+, which is in Benedicts, and it will be used to form copper 1 oxide. So copper 2+ is going to copper 1+, and copper 1 oxide is insoluble, and it will give rise to a color change. Now it's noteworthy that this solution is alkaline, and this works under alkaline conditions. If we have a small amount of reducing sugar, we will get a small amount of copper 2 oxide, so the color change will be different. So low amounts of reducing sugar will give a color change of blue. Moderate amounts, green or pale orange. Red or dark red will mean you've got a high amount of reducing sugar. So these color changes can give you a qualitative indication as to the amount of reducing sugar. However, there's no quantitative test here. To quantitate the amount of reducing sugar measured, you will first need to produce a standard curve, looking at color change using absorbance in a colorimeter, and you would test known concentrations of a particular reducing sugar. Then for unknown samples, you would read off the absorbance and you would use your standard curve to measure the concentration. For non-reducing sugar, this will not automatically react with Benedicts reagent. And it's important to note here that when adding Benedicts reagent, you need to add 5 centimeters cubed, roughly that amount, and you would need to warm it, let's say to about 40 degrees Celsius for about 5 minutes until you get a color change. Now, if you add non-reducing sugar, there'll be no color change. That is because sucrose cannot reduce copper 2+ ions. So first you need to prepare this sample by converting it into reducing sugar. To do this, you need to break the glycosidic bond. I would first add 1 molar HCl, and I would add a few drops. I would then boil for 5 minutes. And here I put this solution in a test tube and I put it in a boiling water bath. Then I need to neutralize that acid by adding sodium hydrogen carbonate. I would add it dropwise until I neutralize the solution. I can test for pH 7 periodically using universal indicator paper. Once I've done this, the sucrose would be hydrolyzed to give glucose and fructose, both of which are reducing sugars. And then I can use the method already explained to test for reducing sugar. Biuret reagent is similar to Benedicts. It contains copper 2+ ions and is alkaline. However, this reagent is used to test for proteins. Note that it can't be used to test for separate individual amino acids dissolved in solution, because you need to have peptide bonds for this reaction to work. And if we have protein present, you get a mauve color as shown. Biuret and Benedicts both have a blue color because of the copper 2+ ions. So if there's no sugar or protein present, it will remain blue. Iodine is a molecule that tests for starch. Iodine will contain aqueous molecular iodine mixed with aqueous potassium iodide. Now it's known that when you mix these two together, you form I3- ions in solution. These ions interact with the starch, and through a mechanism that is not fully understood, this leads to a reaction which produces a blue-black color. So here, iodine, which is going to be orange or brown, we result in a blue-black color if starch is present. Final test is for triglycerides, and it's noteworthy to remember that triglycerides cannot dissolve in water because they are hydrophobic. Because of this insolubility in water, we need to use a substance that will easily dissolve the triglyceride. So firstly, we are going to add about 5 centimeters cubed of 100% ethanol to our food or through our sample. This will easily dissolve the triglyceride, and we need to shake vigorously for it to do this. Now, once this is done, we need to now add an equal volume of water. This will reduce the concentration of ethanol from 100% to 50%. Because of this reduction in concentration, the triglyceride will precipitate. It will come out of solution. And so it will form a white emulsion. And the white emulsion is basically the precipitated triglyceride. If the solution remains clear, then it will mean that there's no triglyceride present. Remember, you need to shake vigorously at the first stage, and after adding water, it's also a good idea to shake the test tube. If after adding ethanol, you don't get a clear solution, then you may need to filter the solution to remove any insoluble substances, and then the clear solution you would add water to. In this section of the video, we go beyond the A-level specification. The first slide here, we want to look at what is meant by reducing sugar and non-reducing sugar. What makes it reducing? Reducing sugar, for example maltose, contains 2 alpha glucose units joined together by glycosidic bonds such as this. Now, the glucose units contain what we call anomeric carbons. These are carbons at which we find hydroxyl groups that can be in the alpha or beta conformation. And also we can have at this carbon linear molecules. So you could have a circular molecule here, a ring structure, and at that carbon, the ring structure could break to give a linear molecule. The 2 anomeric carbons that we've got in glucose are on carbon 1. Now you can see the carbon 1 here has a free OH group. This allows this glucose to form a linear molecule. And you can see the anomeric carbon, and now you can see it's given rise to an aldehyde group, a COH group. Because of this ability for the anomeric carbon to switch between the ring form and this linear form, the aldehyde, right, is able to react with copper 2+ and is oxidized to carboxylic acid. And at least one glucose molecule in this disaccharide is able to form this linear structure, allowing for this oxidation. However, in sucrose, which is a non-reducing sugar, the anomeric carbon in the first glucose unit, the first unit of saccharide, which is a glucose unit, the anomeric carbon is circled. However, in fructose, the anomeric carbon is here. You can see they are both involved in glycosidic bonds. There are no free OH groups. Therefore, this cannot react with copper 2+. if we convert this into two monosaccharides by hydrolyzing that glycosidic bond, the resulting beta fructose and glucose will both have anomeric carbons. They can both be converted into a linear form with an aldehyde group, which can react with the copper 2+. And therefore, we have this principle. If the aldehyde group is oxidized, it will in turn reduce the copper 2+, giving rise to a color change. So how does Biuret reagent work in detecting proteins? Well, here we see we have two polypeptide molecules. We have one here and another one there. And you can see there's 4 nitrogens as shown here. And those 4 nitrogens are forming coordinate bonds with the central transition metal ion. This is known now as a complex ion. Transition metals, like copper 2+, can form coordinate bonds with atoms that have lone pairs of electrons. So here I have 4 nitrogens, each one has a lone pair of electrons, and because the electrons are negatively charged, they are attracted to the positively charged copper 2+, forming these coordinate bonds. And the whole structure is referred to as a complex ion. Now you can also see that these nitrogens in the structure on the left are part of the polypeptide chain, and they are forming peptide bonds. So the NH group here of one amino acid is forming a peptide bond shown there to the carboxyl group of another amino acid. And this peptide bond is also seen at the other nitrogens as shown. This is important for complex formation. Copper 2+ ions, which are normally found in solution in water, form coordinate bonds with water molecules. And this gives rise to the characteristic blue solution that we find in Biuret and Benedicts reagent. And so, if we replace the water molecules with the nitrogens from the protein, then you will see that the complex ion that we form is going to be very different. This does result in a color change, and this complex here is going to be mauve. I hope you found this useful. Please feel free to check out my other biology videos on YouTube and also on Facebook.